All right. Hello, everyone. This episode of the podcast, I had the pleasure of talking with Dr. Laszlo Boros. Laszlo is an expert in deuteronomics, or the study of deuterium. He spent 21 years as a professor of pediatrics at UCLA in Los Angeles School of Medicine. And there he is the co-director of the Stable Isotope Research Laboratory and investigator at the Clinical and Translational Research Institute at the Harbor-UCLA Medical Center. As I mentioned, he's an expert in deuterium, which we discussed in depth in this episode, so it's pretty technical. I try to keep it high level. This is one of the most under-discussed aspects of health, in my opinion, and I'm really excited for you to get educated on this topic of deuterium. We have a few more podcast episodes coming that are also going to cover this, so just wanted to introduce it to you with probably the world expert, or one of the world experts in deuteronomics, and that's Dr. Laszlo Boros. This is Decentralized Radio. I'm Tristan.
And I'm Ryan.
The goal of this podcast is to help educate you on how to live your most optimal life. We will host industry expert guests to shed light on topics that matter.
We are not gurus, rather two individuals who have had to pave their own path to health and vitality, independent of the centralized systems that plague modern society.
All right. Hello, everyone, and welcome back to another episode of Decentralized Radio. Today, we have an exciting podcast ahead, one that I've wanted to do for a bit of time now. We're going to talk about deuterium with Dr. Boros. Laszlo, how's it going? Thanks for coming on.
I'm very good. Thank you so much for inviting me. And you are in Australia. I'm in Budapest. So it's an intercontinental talk for that matter.
Yeah. It's been fun arranging time zones here the last couple of weeks with podcasts, and yeah, it's cool to kind of be around the world and connect with everyone. And like I was saying pre-show, this is a topic that seems to be gaining momentum. And it's funny because I was reading a bit of Gabor's book, Defeating Cancer. And I didn't realize that was like 20 years old when, he wrote that. And a lot of this research has actually been going back a few decades on deuterium. So I guess at first is maybe your background. I know you're at UCLA for quite a bit. And how did you get interested and involved in really becoming one of the world experts on deuterium?
Well, it's kind of a funny and interesting story. And that is, indeed, I got my medical diploma in 1987 from the Saget School of Medicine, where Albert Szent-Györgyi received or worked for longer periods, and got his Nobel Prize in 1937 regarding vitamin C, fumarate hydratase, and biological oxidation. In fact, my grandparents knew him back then in the late 20s because they also lived in Szeged. And after medical school, I got a domestic fellowship from the Hungarian Academy of Sciences, but I was only working for about a year and a half fulfilling my fellowship. And then I left Hungary and I went to Germany. And then from Germany, I went to the United States, the Ohio State University, Columbus, Ohio, where I spent 5-6 years as a research scientist. And in 1998, I joined the faculty of UCLA, Department of Pediatrics, Harbor-UCLA Medical Center. And I started working with Dr. Paul Lee, who at that time was one of the groundbreaking scientists in stable isotope chemistry and stable isotope metabolic profiling. In fact, later we started a company offering stable isotope metabolic profiling, stable isotope-based metabolic profiling services for academics and pharmaceutical companies. And then I became a professor of pediatrics of the adjunct series in 2017. And then I started working for medical and scientific journals as editor, and I've been reviewing articles for quite a long time. and I retired my UCLA position in 2021. And ever since, I am teaching deuteronomics, which is the biological study of deuterium in the living cells and their compartments, and also in the environment, food especially, and drinks, and so on. And we are learning in a continuous pattern of how deuterium affects almost entirely the whole biological system, and what are the specifics, what are the biochemical background information that we need to consider. And deuteronomics goes back in time to very ancient scriptures, and we are just exploring a whole set of very exciting and very novel yet very old knowledge, and positioning them in various translational scientific arenas in medicine. And I do a lot of editing for scientific journals, including Scientific Reports, which is the fifth most cited journal on this planet. And we do position deuteronomics, or the Human Deutenome Project, into scientific arenas or areas where they can be used both for diagnostics and for therapeutic purposes.
Wow. Well, that's exciting that you can kind of focus your whole career on this right now. And I guess I want to get into maybe what was the inflection point for you to really hone in, but maybe for the audience as well. It's easy for me to just get carried away in this topic, but for those who aren't as familiar with deuterium, why it's so important, maybe we can just give a quick introduction on, what deuterium is, and get into maybe a little bit on why it's bad for our health, or why we're more exposed to it than usual or in the past, and then get back to your work and what you're focusing on now in more detail.
Thank you so much. Well, indeed, I do deuteronomics entirely. That's what I do now, and that's how it's been in the last 10 years. And the story starts back in Los Angeles, when one of my friends who is involved in freighter shipping business, he told me that he is shipping this deuterium depleted water to the United States for therapeutic purposes, and it's very helpful for various diseases. And I didn't know exactly what to make of it yet, because I was not involved in deuteronomics or deuterium research. Back then, it's early 2000s, so maybe it was 2001. And he told me that he wanted me to do some experiments, and he actually brought some of that water, deuterium depleted water, to my lab. And I was not interested in particular in it at that time, because we have to have an agreement, a research agreement for that matter. So I told him I need to talk to the investigator who is studying this and providing, and that was Gábor Somlyai, whose book you read. And sure enough, they were interested in exploring the effect of deuterium depleted water in cancer cell culture studies, and we did very explorative and very fundamental studies using different concentrations of deuterium depleted water in cancer cell cultures. And we learned that actually it has a very clear biochemical effect on cells, cell cultures, especially tumor cell cultures. So we published that data. And for the audience to understand what deuterium is, first we have to cover what hydrogen or protons are. Hydrogen, and the nucleus of hydrogen, the proton, is the simplest element in our periodic table. And also, the way I like to describe this, those are the ping pong balls of life, because hydrogen carries energy, makes chemical bonds, and drive these ATPase nanomotors in our mitochondria that provide ATP or adenosine triphosphate for energy balance purposes in living cells, but also releases energy when it joins oxygen to form metabolic water or matrix water. Two protons would join an oxygen from air, and the protons come from food, or the hydrogen come from food. And when we oxidize, when we burn food, practically is to generate this metabolic water, mitochondrial metabolic water, to provide energy as well. So usually the way we describe this is that there's a chemical reaction behind energy production in our cells, and that's water formation. So when oxygen joins two hydrogens or two protons, around 280 kilojoule per mole energy is released. So that's the bulk of energy in the form of heat that is produced in cells. And in the meantime, these protons, before they join oxygen, taken off of food, mostly fatty acids, they power these nanomotors. They release about 30 kilojoule per mole energy. So after all, when you add these together, you end up with at least 350 kilojoule per mole energy for one water molecule to be formed in the molar range. So practically, it is the most efficient energy producing system. It's a hydrogen powered engine in that matter in our mitochondria. And these are actually rotating nanomotors. If you some of those animations, you can see these are like hair dryers, but the rotor or the rotating part is powered by protons and not electrons. So it's a highly efficient, very critical, basic, fundamental element, and the foundation of life, practically the operation of these nanomotors. They are unique and uniform in all living forms, so all cells are using the same type of nanomotors. They can reach extreme revolutions, about 100,000 rotations per minute in some of the prokaryotes. They also use these nanomotors very intensely for various purposes. But practically, hydrogen is the atom, the smallest atom in the periodic table, which can deliver all this work for our body when it jumps from carbon to oxygen, and during photosynthesis from oxygen to carbon. So life is practically about using protons, oxygen, and carbons to capture the energy of light and to transfer this into living systems where these three atoms, carbon, oxygen, and hydrogen, the most common ones in living organic systems and molecules, that's how energy is produced practically when these protons are jumping from atom to an atom. In the meantime, they rotate proteins or provide structural chemical bonds. Now, as you would expect, if an atom has a stable isotope pair, one proton and one neutron can actually make a deuteron, which is unfortunately twice as large and twice as heavy when we look at the nucleus of this proton and the neutron attached to it. And it makes protons' operations difficult simply because they bind, they replace, they exchange with protons when chemical bonds, for that matter, are available. And they break these nanomotors and they break all moving proteins that protons would power in our system, in our cells. And for that matter, deuterons, which are the stable isotopic pair of protons or hydrogen, they can damage moving proteins and moving parts in our cells. But in the meantime, deuterons are also important in collagen, for example, where you have structural proteins. Actually, deuterons provide this unbreakable chemical bonds in structural proteins that make our body, make our bones, for example, cartilages and tendons, stronger and more durable, for that matter. So it seems like deuterium needs to be regulated in our body, in our cells, to provide a structural element in bones. Those are the most common places. Actually, tumor cells like using them because of their structural capability of making structural bonds and sturdy bonds in proton driven structures. And also, what is really, I would say, critically important, understanding the proton-deuteron ratio or the significance of it, is practically our nutrition, our lifestyle, and our health in general. So it's really, our body is able to regulate deuterium simply based on chemical reactions that are designed to form or make structural proteins, and our cells can actually separate protons and deuterons for various biological purposes. Some of these details are still under research and scientific discovery studies. We do a lot of collaborations and discussions with Dr. Stephanie Seneff, who is involved also in deuterium research and deuteronomics. And the other important aspect of deuteronomics or deuterium compared to protons is that this, what we call is the kinetic isotope effect. That means that if a deuteron, twice as heavy proton, like quote unquote, replaces a proton, then it's going to be 8 times to 15 times harder to remove from that chemical bond, or impossible to remove when we talk about proline, which is an amino acid. If it gets deuterated, that proline amino acid can be cooked in 20% hydrochloric acid for 3 days, and it would not lose the deuteron from its chemical bond. So it's extremely difficult atom or isotope to remove from various structures. And for that matter, our body has to regulate proton-deuteron ratios for physiological, biological, and for health purposes. And in a summary, I think it's a little bit technical, but in very simple terms, practically deuterons are your SUV in your driving, in your driveway, that may not be able to park your car in the garage simply because it's twice as large, twice as heavy if you have a passenger car. So it's practically the twice as big twin brothers of these chemical atomic principles, proton and deuterons. They behave very differently, yet they are twins in that matter because they have one proton, so they are the same atomic kind of structural. And if you look at the nomenclatures, by definition, a proton determines the atom. So hydrogen and deuterons are practically the same atom, but the stable isotopic pair, deuteron, is twice as heavy, twice as large, going with kinetic isotope effects that are in some systems is unpredictable. So, and proton is the most common atom in our system, is the most abundant atom in our system, is the most mobile atom in our system. So deuterons have to be regulated very carefully by our cells for basic health purposes.
Hey, friend. Thanks for listening. If you really enjoy this podcast, it would be really appreciated if you left us a 5-star review on Spotify, Apple, or subscribe to our content on YouTube. This helps us get to a larger reach and a larger audience to spread this wonderful free education. Yeah, that was a fantastic overview. I mean, there's so much I want to dive into there, things like proline, trapping deuterium. But first, for the audience, I think the way I see this is to understand this at a higher level, and you can comment on this, is really deuterium is affecting the mitochondrial output, right? The mitochondria, the electron transport chain, is building up this proton gradient, this mitochondrial membrane potential, this voltage, so that it can pump protons through our nanomotors. And when those protons are deuterons, which are twice the mass because of that neutron, that's going to mess up the process, and therefore, it could lead to reduced ATP production, just reduced metabolic water production, mitochondrial dysfunction, which we know is kind of leading to a lot of chronic disease down the road. So, and there's so many other things to couple that with, and that's why it's cool that, Stephanie Seneff, who we're going to talk to as well, is coupling that with glyphosate. But I want to get into maybe the ATPase a little bit. So are the deuterons, are they like slowing it down, like gunking it up, like kind of putting in lower quality gasoline into a high performing vehicle? Or are they kind of like stopping it completely after a certain point? Because that's something I've been curious about. Is that dependent on just over time? Because obviously, cells are being apoptotic and turning over quite a bit. But I'm curious if there's an answer to that.
Deuterons, they make these nanomotors stutter and they break these nanomotors. Okay. So depending on their rotational speed or the rotations they perform in a minute, and practically deuterons, as they try to fit into the nanomotors, carry 9 to 12 protons at any given time, and they pick them up from the outside of the inner membrane of the mitochondria. Mitochondria should operate in a deuterium-free environment. That's how we get kind of a good description of the situation. Definitely very low in deuterium, the mitochondrial environment, the matrix especially. And Dr. Abdullah Olgun wrote a paper about this, and this is I think the best paper to learn about ATPase and the deuteration or deuterium damage delivered to these nanomotors, simply because there are mathematical calculations and there are good models and systems to apply to study the effect of deuteronation or deuterons or deuteronomics of these nanomotors' function. And you're right. Mitochondria is a very unique place because, as far as we know how nature operates, the mitochondrial matrix or the intermembrane space is what can separate protons from electrons and produce these positively charged proton hydrogen nuclei that are very reactive. They explode in nature. If you smell gas in your apartment, the first thing you need to know is don't turn on the light switch, because if that little spark would activate oxygen, the whole apartment building kind of explode. And that's a hydrogen-oxygen. The same reaction happens in mitochondria under controlled circumstances. And that means these reactions have to be controlled and occur in a very controlled and slow pace to release all the energy. And the nanomotors are part of this, controlling the proton flux or the proton flow into the mitochondrial matrix, where activated oxygen is waiting on complex 4. You probably read about the protein complexes of the electron transport chain, which provide a negative charge, kind of the spark plug for this whole system that activates oxygen. And oxygen and hydrogen, when it unites, that's when the bigger energy or the explosion, quote unquote, in biological systems, they don't explode because these systems, these reactions are controlled. But that's practically how most of the biological energy is generated in mitochondria and deposited into phosphate bonds when it comes to ATP, or just released in the form of heat when metabolic or matrix water is formed. And understanding these processes between mitochondria and the peroxisomes, which we have not talked much in the past, but it seems like we have to bring in the peroxisomal beta oxidation or fatty acid oxidation and hydrogen peroxide formation and metabolic water formation into this whole system. And this is the newest, most recent paper we wrote, and it's under review now. And we kind of approach the system of the Himalaya, Mount Everest climbers who can do this without oxygen, supplemental oxygen. And it seems that they have to utilize these nanomotors, these metabolic water forming systems, and their peroxisomes using dissolved and also carried oxygen, oxygen that is carried by hemoglobin, and also using the dissolved O2 or oxygen gas in our blood to be able to climb all the way up to the Mount Everest in ketosis, in fat burning state, in a deuterium depleting fat burning state. It seems like that's the key to achieve any extreme sporting challenge, or like, for that matter, climbing up to the top of the Mount Everest, the Himalaya. Mount Everest is I would say that's the ultimate challenge without supplemental oxygen. And you can do this if you understand the deuteron, proton, energy production, nutrition, kind of connect those dots, and it's possible, but you have to kind of learn the biochemistry of it. And this is what we did. But it seems that after all, these nanomotors that stutter and then break in the presence of deuterium, that's why we have to be careful what we eat, because the deuterium content of our food will determine the deuterium content in our mitochondrial matrix and the nanomotor functions and the energy balance of our cells. So for that matter, this is the most fundamental and critical principle and steps in biochemistry that we need to understand. Now, I was lucky enough to know Dr. Paul Boyer, who got the Nobel Prize for nanomotor functions. He was at UCLA. We buried him in, I believe it was 2018. He was a great scientist. He discovered these, but the way he described it is that rotating proteins performing chemical reactions, which was huge at that time, late 1980s, mid and late 1980s. And deuterium research came in early 1990s, mostly by Dr. Gábor Somlyai, who showed the first time in Budapest in Hungary that deuterium proton-deuteron ratios determine biological behavior and cell transformation. And from then on, he could not connect this with metabolic water production. That was my biochemical contribution to this whole process. And ever since, we saw more and more scientists applying deuteronomics research or the Human Deutenome Project to their particular field and area. And there is now a growing set of publications that, especially in oncology or experimental oncology, and to some matter, clinical oncology, where we actually determine deuteron or deuterium levels in the patient's breath, and we make clinical translational predictions and outcome measures. And there are increasing numbers of publications in the peer-reviewed medical literature that use deuteronomics in their discussions to explain their findings.
Yeah, wow. I mean, it's so cool. Just the fact that, the nanomotors getting into that and how much progress they're making. I'll have to link that paper by Abdullah Al-Ghun. I was reading that actually earlier today in preparation, and it's fascinating, and especially getting into the half-life of these nanomotors, the F0 subunit. I think it's like 40 to 50 hours. So just thinking on the scale of these mitochondria is really fascinating to me. But you mentioned a lot of important parts there, right? And kind of what I want to get into is why we might be, having more deuterium in our environment, and thus higher deuterium being produced as a result in our mitochondria. And a lot of that's due to the food we eat. So from my understanding, it's basically that the carbohydrates, as you mentioned, ketosis being important. So the carbohydrates are kind of highest. The processed foods are highest in deuterium, and then the fats are the lowest. And I think it's spanning, quite a bit. But before maybe even getting into that, what is, normal levels? So from basically what I've read, like 150 parts per million is pretty normal. Is that now, or is that something as a result of like modern society? Like that's what's common, maybe not normal. Maybe normal is like 145. So maybe you could shed some light on that, and then we could get into some of the food aspects of it, because that's really what I think is cool.
Well, a normal is hard to determine, because if you look at anthropological data, and if you look at historical data, and you look at geographical physics data, you learn that because of the temperature changes of our planet, of Earth, the biggest source of water vapor, and for that matter, deuterium is carried in our oceans in the form in water, and currently the amount of deuterium, the concentration is 155.6 parts per million, which means that out of 1 million hydrogen atoms in water, 155 of them are deuterons.
And now, that's in ocean water, correct?
That's in ocean water. It's called the Vienna mean oceanic water deuterium concentration. That's what laboratories use
as the baseline,
as their standards, as their baseline. This is the most abundant deuterium, environmental deuterium that we can be exposed to. Maybe there are a few ppm increases when you go to the Dead Sea with higher elevations and high evaporation rates. Maybe you can measure in some environmental water 160 ppm, but it's between 155 and 160 ppm in the environment, even under extreme conditions. Now, you can find lower deuterium contents easier based on historic, geographical, and also anthropological data. Back in in cavemen's time, 15,000 years ago, they did a study of oceanic vapor captured by volcanic eruptions and deposited in those ash deposits. The vapor, the oceanic vapor, deuterium content was only 135 ppm. So as we move throughout the history of our Earth planet, based on temperatures and some other factors, winds, and so on, you see numbers between 135 and 155 ppm, which is 20 ppm. It's quite a lot when you kind of look at the physics of deuterium. And in fact, in animal samples, you can see these variations, and we believe that the human, the anthropological aspect of it is that the time of creation or the time of evolution, you use the way you want to use it. I'm not using evolution very much, because there data contradicting the process now. Even science cannot prove that with the development or advancements of genomics research. So it seems like it's a dead-end road explaining these biological systems. So I would just use anthropological adaptation. We were adapted to lower deuterium environmental, lower environmental deuterium exposures, maybe in the range of 135 ppm. That was the oceanic vapor, and the vegetation was using as rainwater a lot lower, about 20 ppm lower deuterium concentration, and this is how biological systems evolved. Now, prokaryotes, which are the bacteria and mostly, ferment sugars, they do use deuterium to make their DNA unstable, so they continuously provide a dividing force, meaning that a signal for continuous cell divisions, and this is how cancer cells behave. But it seems like it's a deuterium-based process to keep DNA unstable and chemically in an unemployed state, meaning that it's never kind of stops growing, the DNA content, and the cells have to divide continuously. That's very characteristic of bacteria. They cannot really wrap their DNA into a nucleus, meaning that they cannot really show the morphology of eukaryotic cells who have mitochondria are able to deplete deuterium and form a stable DNA that can be locked into a nucleus, and they only divide in case there is excess deuterium in their environment, or they divide based on various growth signals. But it seems that the biological behavior as far as prokaryotic or eukaryotic cells or organelles, those regulate these phenotypic behaviors or these behavioral patterns by the deuterium that they recruit from the environment, from sugars mostly. If you look at bacterial growth, they always grow in sugars and not on fat. If you leave your bacon out in the sun and an apple piece next to it, in a few days, your apple will be rotten by bacterial and fungal fermentation, but your fat may be the same practically because of the low deuterium content. Fatty acids are not a good source of deuterium for these continuously dividing cells. And for that matter, in fact, understanding the process of how deuterium is depleted in fat kind of explains how diet, from the dietary perspective or from the ketosis perspective, why our sleep patterns, why when we are born, why we actually operate in the ketogenic metabolic state that helps us to regulate deuterium for eukaryotic cells so they can behave in a differentiated pattern or manner, and they can contribute to determine organ functions. And when deuterium accumulates in these cells, nanomotors are broken, metabolic crowding occurs, meaning that you cannot really get rid of the carbon source or the carbons that you take in the form of food, organic molecules that you burn, and practically you develop chronic disease processes, obesity, diabetes, various dementia-related conditions, metabolic syndrome, cancer is a big deal nowadays. These are most like chronic epidemics. I'm working with the Kennedy team and other medical teams on deuteronomics, and the chronic disease epidemics in the United States is a huge. It's really a huge task even part of presidential debates, simply because back in the 70s, we looked at 6-7% of population having chronic disease processes. Now it's close to 60 or even over 60%. So it's, 7-8-fold higher than it was just a few decades ago, 3-4 decades ago. So this needs to be addressed from the fundamental basic biochemistry and medical research aspects of those are very critical to address these devastating trends in human health and life expectancy. Now we can see changing, deep dropping in the United States. So it seems like the food industry, the processed foods, the carbohydrate rich dieting, the low fat products, whatever those are, I tend to eat in a carnivore lifestyle using grass-fed animals. And thank you for covering some of the papers we wrote. Those are very important contributions to understand this deuteronomics to serve your health. And that's practically the best way to control your deuterium intake by the appropriate food and nutritional and lifestyle matters. And we would like to kind of position the normal range as we see biochemically, that should be probably close to the 130 range instead of the 150 or 155 range, which is unfortunately very common nowadays when we measure deuterium concentration in breath, for example, vapor of exhaled human breath. We would like to see those in the 130 range.
Wow. I think that's so cool because it sounds like it's not that big of a deal, like 135 to 150 or 135 to 155, like you're saying. But I know that's, a tremendous difference in terms of health and mitochondrial function, right? So basically what you're saying is that through majority of human existence, our biology has basically been designed to operate at around the mid 130 range parts per million. And it's only been recently that we've kind of had this warming. Is that really the main cause, or is it just the variations of sea temperatures and everything
industrial
combined?
Processes when it comes to food production, the GMO,
I just meant from the water perspective.
The water is temperature, wind, and solar and light electromagnetic exposure. So it is a physics related factor. How the water deuterium or the water vapor, the water is always almost the same. It's 155 ppm. The vapor of it that provides the rainwater for vegetations, that's what determines the deuterium environment. Because we don't drink oceanic water, and even the animals that live in the ocean, they cannot drink oceanic water because of the high salt content. Yep. And I think that's purposefully, that's how it was created. But after all, indeed, we depend on water vapor for vegetation and biological response from the dry land of the vapor that comes from the oceans. And that determines, and obviously various crop-related industrial processes can change fundamentally the deuterium discrimination properties of these plants and growth rates and so on. So this is what we see. Now, reflecting back to your question, you need to look at deuterons or deuterium as not as their abundance is a problem, because it's really not an abundant isotope. It's one of the least common isotopes. It's a lot less than C13, which is the stable isotope of carbon. But the carbon isotope, which is 1.1% of all carbons on this planet, instead of 0.015% when we talk about deuterium, so that there's like magnitudes of differences. But the stable isotope of carbon is only 8% heavier than the parent ion, or the parent atom, carbon in that case. It has a molecular atomic mass of 12 Daltons. And proton has only 1 Dalton, and deuteron has 2 Daltons. So it's 100% increase. It's doubling the size, and it's a huge change as far as increasing the weight of an isotopic pair. So the damage that they deliver, that's huge. The damage that they can deliver in a biochemical system, that is devastating. It's the way I usually describe it is that just look at an airport where there are so many people changing airplanes and coming in and out, and all of a sudden there's a terrorist appear somewhere. Somehow they saw somebody running, and they shut down the whole airport simply because they have to find that one guy, instead of just kind of letting happen everything and see if they can see this guy in action. And then they start looking, they shut down the airport, and until they find that guy, until your biological system finds that deuterium, it shuts down or tries to adapt to the situation where there is a deuteron somewhere that is not belonging there, and or if it makes it there, then your biological system will respond in a dramatic way. So even though it's an uncommon stable isotope and the differences 20 ppm may not be as large or significant from like the bird eye's view, when you look at the very rapid rotating nanomotors, even changing 1 or 2 ppm in deuterium, because of the velocity of these nanomotors, the likelihood of these nanomotors to being broken and kind of overloading your deuterium scavenging mechanisms in your system, that risk is increasing. And when that happens, when this scavenging mechanism is not sufficient, then cellular damage and mitochondrial damage is imminent, and chronic disease processes are imminent. So it's practically the biological damage that deuterons can deliver. Those are more like an elephant or a bull in a China store, China shop, simply because they just destroy all moving proteins, especially rapidly moving proteins. And for that matter, their amount, their concentration needs to be very tightly regulated in the physiological range.
Yeah, so that even a 1, 2, 3 ppm difference is pretty substantial, basically, is what you're saying.
Yeah, so just give you an example. If you talk to a physicist, they're going to give you the deuterium concentration in per mil value, meaning that they are not using percentage. They use per mil, meaning that they use 1,000 protons and measure the deuterons among 1,000. It's not in 100, it's 1,000, because they have to measure this so sensitively, because it has such a biological impact that they have to multiply the scale that they use to measure this by 10, simply because they want to know even those miniature changes are so dramatic and so important in determining the age of a biological sample, of determining a water source from different geographical or geophysics phenomenon. That's how they determine the source of drugs, for example, where they are from. These stable isotope patterns are very descriptive, meaning that very characteristic of the biological or physics or the source of those organic materials. So practically 1 ppm is if you talk to a geophysicist, is such a big change that they would actually use a different scale to actually even kind of tune finely into that change, simply just to understand more of the physics or the reactions that contribute to those changes. And it's quite an interesting and intriguing science, deuteronomics, just to see this stable isotope, how it behaves on our planet, and obviously other planets as well, because that's how they use. That's how they determine water source, intergalactic atomic compositions. If Mars can be populated by humans, deuterium is a huge factor, because there are more deuterium in the ice caps of Mars, as I understand, and they actually measure deuterium in Saturn, water, and the moons. Practically, it's if you look at not only the biology but the physics and the biophysics of it, it's really a very influential.
So it seems like they're pretty aware of it. Then, like, why has nobody been talking about this in the mainstream? If they're looking for this on other planets, are they just making sure that it's not like 200, 300 ppm in the water or something like that, or?
Well, because you can't make much money out of it. It's okay. Everything is money driven. They don't care as long as it's just some. First of all, it's not like financially very very beneficial operation to take something out instead of adding some like a supplement. Cost this much or that. If you
pharmaceutical.
For example, it's a very different kind of mindset, and we are on our own. I mean, you guys run an independent radio, so we can cover, I guess, all kind of aspect of it. But you need to be aware of that, especially in medicine, pharmaceutical operations. They want to sell something to you, meaning that it has to be a supplement, it has to be a drug, it has to be something that they can put in a box, they can label it, all kind of fancy stuff, they can take it to the television, they can advertise it for you, and then you're going to buy it. Deuteronomics or deuterium depletion is not like that. I can't really give you a pill that will deplete your deuterium, but I can give you some ideas where to buy your meat source, where to buy your food source, what to drink, how much to drink, why to drink, when to drink or eat, and how to sleep, what kind of exercise patterns, what kind of nutritional protocols, what kind of lifestyle you have to pursue to actually control your deuterium in the physiological range and not to get sick, for that matter.
Yeah.
So it's a different kind of mindset in business.
Yeah, that's the mindset we have right here on this show and everything I talk about. It's crazy and it's sad. And I'm curious to get your opinion maybe on the funding of research. But before we talk about that, let's talk about the food a little bit, because that seems to me like the biggest gains people can make. And it's funny because it's right in line with a lot of what's kind of gotten a lot of traction, right? The keto carnivore movement has definitely proliferated in popularity. And it's funny because, nobody's talking about deuterium really at the diet level. And I've done some YouTube videos on, hidden benefits of carnivore and keto that no one's talking about. And definitely deuterium depletion is one of them. And it's pretty substantial. So I think it's a combination of your work, Boros' work, but from what I've read, from eating kind of a low carb diet, you can make mitochondrial water as low as 118 ppm, and then if you're eating like a Western diet, it's not even going to be that deuterium depleted at all. It's going to be like 150, 155.
Are you interested in 100% grass-fed, grass-finished bison meat? I'm excited to be a partner with Falls Family Ranches. Based in Wyoming, Falls Family Ranches is raising high-quality bison meat the way nature intended. As a native large ruminant of North America, bison is one of the most nutrient-dense foods you can consume. If you're interested in trying out their bison boxes, use code Tristan, Tristan, 10 for 10% off your first order. Is that correct?
So that's right. So fat or ACL chains, just to serve both the common kind of lay audience and also, people who are scientifically more interested in this whole process. Fat is low in deuterium because fat or fatty acids are produced from citrate, which is formed in mitochondria in low deuterium environments. So naturally, all fat in natural environments, natural feeding habits, grass-fed and also carnivore lifestyle, the fat part of your food is also low in deuterium. So when you burn it into metabolic water, it's going to provide you low deuterium metabolic matrix water. And that is necessary for your mitochondrial health. Besides that, fat is more saturated with hydrogens per carbon, meaning that it produces twice as much heavy hydrogen or twice as much metabolic water than what you can produce from carbohydrates. Now, carbohydrates are produced from various other precursors, especially in plants during photosynthesis, and there is a big difference between fat and carbohydrate deuterium sources. And Gabor Somjai published a paper, I believe, a year ago, where actually he he reports food deuterium content. And our collaboration was so fruitful simply because we could determine the source of deuterium depleted water in biological systems. And based on the nutritional and fat-based dieting habits, we could predict what kind of deuterium depleted water your mitochondria will produce. And we could link this scenario with health-related issues or therapeutic efforts.
A quick question there. How correlated is that? Like if I eat a piece of grass-fed meat that's like 130 ppm, is it just going to produce like 130 ppm metabolic water, or is there kind of a variation? Because that was my kind of only thing that I wasn't sure about. Obviously, it's very correlated in terms of how low and high it is.
Yeah, so what you want to eat is the fat part, fatty meat, at least 50-60% fat content, and the rest is proteins and very little carbohydrates. In the 118, 110 ppm range. That's the grass-fed natural butter and so on. Gabor's paper have this data. And that's what can actually supply deuterium-free metabolic water in your mitochondrial matrix, because your glycolysis and your biochemical reactions can get rid of this much of deuterium, 118 ppm. I'm just giving you a number. It's probably needs to be below 120 ppm. In grass-fed cow fat, tallow, ghee, those are in the 110, 100 ppm range. So from this grass-fed natural fat source, you can produce deuterium-free or very low deuterium, a few ppm matrix water, because your body has glycolysis and isomerases that can actually scavenge, that can actually get rid of deuterium from the fat source, fat-related intermediate metabolites, and also from carbohydrates somewhat. And because of these ratios, the low carbohydrates are low deuterium, it practically preventing deuterium to entering your system. So it's not the deuterium content, but how much of high and low deuterium containing substrates you consume. What's the ratio of carbohydrates versus fat in your food? Practically that determines your deuterium load. And what's the water deuterium content that you consume. Now, if you look at anthropology, for 4 million years ago, and this was a big finding for me, papers that published that actually the prehistoric man was able to open the school of large plant-eating animals and start eating bone marrow fat. That's what humans were eating and conserving for themselves for millions of years. And evolutionary, if you want to say it, or how the creator planned this, is practically these prehistoric men were able to go in a ketogenic, deuterium-depleted diet, which provides nutritional ketosis and metabolic ketosis with low deuterium, and they were able to evolve or adapt to low deuterium biological habitats or faunas, if you will, and they were able to develop brain skills, fine motor movements, fine kind of hand speech. They were able to use their body in a low deuterium environment in a more flexible, more expressive, and in a more complicated, complex way in as far as memory, as far as communications, as far as social behaviors, and so on. And that was all dependent on low-fat, ketogenic diets that they were obtaining from large plant-eating animals' carcasses. Now, the other advantage of this is that they didn't have to chase, they didn't have to hunt, they didn't have to be exposed to predatory animals by competing for these preys. They just waited till the predators left and the scavengers left, and the bones were kind of cleaned up for them just to break through those bones using stone tools. That's what they found in this Ethiopian land, where they found these 3.8 to 4 million years old carcasses, bone structures of large plant-eating animals that were actually broken into using tools. So if you look at for example, a mammoth, or if you look at large plant-eating animals, they have 20 kilograms of bone marrow in any of those big bones. So those were actually very reliable, good, untouched food sources. They just had to learn how to use tools to get to this ketogenic, low deuterium fat source. And the predators left because they were not interested in the carcass. The scavengers left. The ancient kind of the cavemen's hyenas and the tigers and the predators left by then, and the prehistoric humans were left with these carcasses and a lot of fat, a lot of deuterium depleted ketogenic food source. So as our natural metabolic profile is ketosis, they could maintain ketosis during the day, low deuterium ketosis. This is what we should do, by the way. This is our natural metabolic state. And they were able to adapt to various tasks, and they were able to solve various tasks and form more complex social patterns without being threatened by or competing with predator animals. In the meantime, agriculture came along about 10,000 years ago, and they started cultivating plants that are higher in deuterium. They formed larger communities, and they started harvesting plants and started eating plants. And that's where chronic diseases, and that's where diseases appeared in mass scales, as we know. And this is when infectious diseases appeared also, because infectious diseases also depend on deuterium for the propagation of infectious agents. And changing diets, changing dieting behaviors changed the disease landscape on mankind and societies, and we ended up where we are now. We practically, well, I'm not, but most of the people eat processed food. And if you look at the kind of the general health or the chronic disease epidemics taking place on this entire planet, you can tell there is a huge devastating change. And practically it's because of the environmental exposure and also the food they don't eat, or the kind of food industry does not measure deuterium. They don't really label deuterium, and they don't really care about deuterium, meaning that practically you're left alone. You have to figure out yourself where to find low deuterium food source, and those are grass-fed animal fat. Practically, we are scavengers. If you look at ancient writings, when Noah walks off the ark, Lord tells him that eat meat. When you look at Cain and Abel's story, Cain, the plant grower, kills the animal. The guy who produces animal nutritional kind of guidelines and grows animals and raises animals for their own fooding and providing this for the Lord as a sacrifice. And when you look at the Acts of the Apostles, that you can see that the Lord provides a whole plate of meat. So practically changing this in ancient writings, you can actually go back to more like the historic source of this, or the ancient knowledge of this, as how it's described in these ancient books. But for our current times and going back in anthropology, you can see the overarching trends in changing dieting, changing food source, changing agricultural processes, and human health in general. And by now, you can see the devastating trends simply from going from natural grass-fed food sources to industry-based GMO crops and animals that are actually fed with these GMO crops. The animals are sick, humans are sick in general, unfortunately, and it seems that it all depends on nutrition, it all depends on mitochondrial matrix functions, and it all depends on deuterium loads to break those nanomotors and start these metabolic crowding process that is practically the fundamental biochemical event behind all these undesired epidemiological chronic disease epidemics and the deterioration of human health and life expectancy after all.
Yeah, I mean, I think it's spot on, right? I don't have any qualms with that, really. I think everyone's pretty much in agreement that so many people eat way too much junk food, processed food, and even the carbohydrates, right? The carbohydrates today, they're not even in the fruit form, let's say, they're not really indicative of fruit that would have existed even 100 years ago, 200 years ago. If you go and pick like wild berries or something, they're very, very low in size and thus probably lower in deuterium content as well. So the only question I have is because I've recently interviewed Michael A. Crawford, who's an expert in brain evolution and DHA fatty acids. So his theory is basically that we evolved by eating lots of seafood and the ruminants, like that combination of high DHA, the vital minerals from the seafood, as well as the vital nutrients from land ruminants. So my question is there a substantial difference in saturated versus polyunsaturated fatty acids in terms of deuterium content, like say DHA versus stearic acid, for example?
It could be, and there's a French group scientist. They measure deuterium content in saturated, unsaturated fat of industrial and also natural sources. So you can find some original data. If you read our paper in medical hypotheses about the about the proton exchange reactions in the Szent-Györgyi-Krebs cycle, then you can see these scenarios, how they play out. But practically, once fat is generated, saturated or unsaturated, they are in the range of the lower deuterium kind of arena. So it's below 120. I'm just giving you this number comparing to carbohydrates. When you look at carbohydrates, they are all above 135, 140 ppm. So you're looking at a gap of 10, 15 ppm between proteins, carbohydrates, and fat. And based on what plant or what animal is producing that particular fat and where it is produced, there could be another 15 ppm differences, but it's below the 130, 125 range. So you go from, if you look at carbohydrates, you go from 138 to 155. If you look at fat, you go from 125, and that's the highest end, down to like maybe 95 when you look at some of those, truly natural-based, grass-fed, saturated, long-chain animal fat sources. So
is the number of carbons, like you're saying, the long chain, is the number of carbons kind of more correlated with lower deuterium then?
Well, actually,
I know it's correlated to higher ATP production, right?
Yeah. So the way we kind of differentiate fatty acids, there are short chains. Those are 4, 6, 8 carbons. There are medium chains. They go from 8 carbons to 14 carbons, and there are long chains. There are palmitate, stearate, oleate, and so on. Those go above 20 carbons. So the cut is not that clear. So there's short chains, 8 carbons, and there are medium chains like 16 carbons and then above. And longer the chain is, that means more acetate units. These are two carbon units. That's how fatty acids are built. And from fatty acid synthase over 16 carbons, fatty acid chain elongase takes place. These are enzymes that produce these fatty acids. And they operate using different deuterium scavenging and deuterium capturing or deuterium discriminating mechanisms. It's a very complex biochemical kind of scenario. That's why I don't want to go into details, but practically you go, actually in Szeged, I gave a few talks about fatty acid synthase back in 2014, where actually I described these processes, but it's in and between those 95 to 120 ppm range when we look at fatty acids, saturated long chain, or saturated, unsaturated fatty acids. They are in between those two numbers somewhere, and the specifics you kind of have to measure, and then you have to think over the biochemical reactions that contributed to those fatty acids. But if it's a natural source, grass-fed, or natural source fatty acid that is synthesized from grass-fed animals or modified in our system, because that's what peroxisomes do. They modify these fatty acids, they align them, the liver assembles these triglycerides, and there are low density, high density as far as their deuterium, as far as their protein content is, and or lipoproteins as how we characterize them. But practically every cycle changes the deuterium content, but it cannot be coming from a sick animal that was put in artificial feeding protocols, and even their fat content is 135, 140 ppm. So it's not any different than eating carbohydrates.
Yeah.
So
the grain-fed animals, obviously, that makes a ton of sense. But I'm curious from DHA from an Alaskan caught salmon versus
soybean
those are
oil,
actually good
right?
low source of deuterium. Yeah. Those are salmon
soybean.
the salmon we measure. We have a large database,
Okay.
which I do consult on my website, and you can kind of I can look into these. Say it again.
I'll have to dive down that. I've looked for it before, but
I can go into this database. I cannot share it because it's practically a discovery process based on specific interests. But we can kind of consult on these topics because those are very fundamental and very important knowledge.
Yeah, it's just like polyunsaturated fats have gotten a really bad rep, and they've been grouped together, but it's like DHA from salmon is not the same as linoleic acid from soybean oil, and I bet that has way higher deuterium content. So that'd be cool. Any
we measure those actually, so we are looking at data here. We have original isotope ratio mass spectrometry measurements. And we do have those numbers, but they always have to be put in the context of like what the nutritional goal, what is the amount, what are the ratios, what is the source. Because the problem is if you buy, for example...
meat especially. I mean, where is it from? How was it raised?
Yeah, exactly. So the problem with fatty acids is that fatty acids can go into your mitochondria directly through the carnitine acyl transport chain. It doesn't have to go through glycolysis.
Yep.
Glycolysis is practically created as a set of reactions to check each and every proton on a carbohydrate, on a sugar, on a glucose or a fructose molecule that you consume using fruits or eating fruits. Those reactions of glycolysis is going to remove every hydrogen, every proton from your carbohydrate and replace it with proton that is coming from your metabolic water or cytoplasmic water. It's practically a proton exchange, set of proton exchange reactions. That's why glycolysis takes 10 steps.
Yeah.
To check
That's
every
why the TCA's Krebs cycle is so intricately detailed.
Yeah, that starts the exchange process. Now, carbohydrates have to be checked before carbohydrates source pyruvate or acetyl coenzyme A joins the TCA cycle. It has to be ripped off of all deuterium there is, as long as it's not too high, meaning that it overflows the system. And that's what happens in grain-fed animals. Even their fat content, even their fat is not low in deuterium enough to preserve or prevent your mitochondria to break. And fatty acids shoot right into the mitochondria if they are 18 or 16 chain length, and through the carnitine transport system, and they start burning in the mitochondria and loading mitochondria with deuterium. And there is chronic disease, metabolic diseases, metabolic crowding. And for that matter, you have to know the source, these numbers, and be in the safe zone or the same safe range when you consume fat, because those can actually shoot right into your mitochondria. Those are like when you go to the airport, the diplomacy kind of booth is over there, and those guys they just flip their thingy and they just shoot right through that. Those are fatty acids. And, we as like passengers, we have to wait the line and wait in line. So that's the difference between carbohydrates and fatty acids, how they shoot into the mitochondria. So that's why you have to be very careful what kind of fat source you consume.
Yeah, it's so interesting as well, especially if you dive into just like the adaptation of the mitochondria from like a higher FADH2 to NADH ratio. But we don't need to get into that. So the diet.
These are very good arguments. I'm sorry. These are very important, very critical, very good arguments. So keep reading. You have questions, call me, write me, and we'll dig it in.
Well, it's fascinating to be on the frontier of this research. And I think from the food perspective, it's really simple. It's what, we preach, right? Like eat local, high quality
animal foods as the center of your diet, and local, especially important, seasonal, but you can verify the quality, most important. And, carbohydrates, you limit them. Obviously, if you eat local and seasonal, they'd probably be only available certain times of year anyway. But more importantly, something I wanted to ask, something I've been curious about is dairy, because I've heard mixed things. Raw milk has become really popular in the alternative health space, the primal health diet realm, and I've heard things saying that milk, because it's given to for growth to infants and babies, is actually high in deuterium content, and this is an argument that why we shouldn't consume it. But then I've also heard the opposite. So no better person to ask than you, and this would probably be the last food-related question.
Well, again, dairy has
obviously
a special...
raw pasture milk is probably really different from pasteurized feedlot grain-fed milk. So,
yeah, it's. I'll tell you, this is actually a very good question, and I get this simply because. After all, it's very fundamental and it clarifies some very basics of deuteronomics. And that is, it's very unusual that any species would drink the dairy of another species, because it only has one purpose, and that's practically just look at humans. If you have a baby in the first year of their life, using and eating just breast milk, they actually triple their body weight. They go from 3 kilograms to close to 10 kilograms. And it takes deuterium to develop those structural proteins, collagen, grow bones, make him stronger, those tendons, so in the growing body, in the developing body, and the continuously dividing cells and differentiating cells, deuterium has a special role. And so is the food, has a special role in supplying and providing those needs for physiological purposes. After you pass your suckling age, or you kind of wean off of this, you start eating, animal-based food or food products. For example, what your mom is cooking for you. First just to do those little small cut pieces of meat and fat and so on. This is how we were raised. This is how we grew up. Those little pieces of sausages, those little thingies on your plate, and the babies are learning how to eat those as well. Then you stop drinking milk for them, because it becomes insufficient to supply the energy and the structural needs for it. So you have to start eating more substantial. You have to eat more as you grow. So your mom or your if we talk about animals, then the cows, they stop producing milk. So milk is produced simply because they milk the cow every day. So the cow thinks that still has a little calf that needs to feed. So otherwise the milk production would stop. So for that matter, and I'm just going to plug in my computer, I apologize.
No problem.
And for that matter, dairy is not for human consumption. I mean, period. If you look at lions, for example, hunting, zebra horses, and the zebra horse is a zebra horse, a female that is producing milk. The lions never let their offsprings to actually have that milk. Practically, they just direct their babies to eat the meat and fat if they are old enough to eat. It's really unusual for any species to drink milk, especially from cows that are sick already with high cell numbers. There was a grain fad. those were fed with artificial. Actually, cow would never eat soybeans. Practically, they never eat corn. They love corn when it's this tall and it's still green. If it's a green plant, they eat the corn plant. They don't eat the corn. The corn back then was very different than now these GMO grown corns. I mean, if you look at the cow nowadays, for example, a Holstein farm, you go to a Holstein farm, there are industrial kind of, grain fed. Mm-hmm. They only have a lifespan of 5 years. For their last years, practically, they're barely alive. They are very sick, and they maybe have 2 offsprings, and they have 2 calves, and they produce 30, 40 liters of milk a day, and then they have to butcher them because those animals are unable to live any longer. Well, if you look at, for example, Dezső Somor, who is my good friend in Hungary, and he actually raises Hungarian gray cows, water buffaloes out in the pasture. What do you think? How old is his oldest cow?
20 years.
34 years old.
Wow, that's pretty good.
With 25 calves.
Yeah.
Not 2 calves, and those Holstein cows, they have to be pulled off of onto the plateau of a truck and have to be taken into, because they can't walk anymore. That's sad. The milk that you're drinking, and I'm not, but that's what the dairy these days.
Yeah, so mainstream dairy. Yeah,
mainstream dairy. So we wrote a paper about this in Metabolomics, which compares the metabolic and the disease state of the grass-fed and the grain-fed cows based on published data in the literature, and it's in Metabolomics. So if you want to read more about it,
that's the what to feed or what not to feed, right?
Exactly. What to feed, what not to feed.
Yeah. I read that is fascinating. But I
listen,
just had
I'm
to
not
ask
shy,
the dairy.
meaning that.
Yeah, I had to ask the dairy question because the
of course, that's
dairy
very critical, very important
but
question.
It makes sense, right? If you have to grow, obviously, I would imagine the deuterium content would be pretty high in some.
Yeah. Those. We measure those. From your perspective, if you compare sour cream or butter from grass-fed cows compared to grain-fed cows, if you compare those
15 ppm difference, 20.
So you go from 110 to 136.
Wow.
So,
26. Yeah. Wow, that's incredible.
26 ppm. Listen, it's not a joke.
Yeah.
I mean, it's not just something just to walk by and say, oh, well, whatever. It is significant. And when it comes to human health and animal health, or just planetary health, or just practically chronic disease epidemics, you have to take these into consideration, unfortunately.
Yeah, 100%. Well, thanks for entertaining that. I think it's important. And again, it's the quality of the food, the quality of the animal that you're eating matters tremendously. And we even know this proven via metabolomics from the nutrient density perspective. We've talked to Stefan von Vliet on this show. So we now have the data to prove the logical conclusions that the quality of the animal you're eating matters. And now with this deuterium information, it shows you how even more so that matters. But I guess the next piece is that really can make a difference on is the water you drink. And wow, what a weird world we live in, where everyone's going to the gym, walking around with, 2-liter, 1-gallon water bottles, or dead water that's probably super heavy in deuterium and not hydrating their cells at all. Their mitochondria dysfunctional. They're not making the right metabolic water. So I know, listening to your podcast with, I think it was Dr. Anthony Chafee, you said that you pretty much never you drink as little water as possible. And then when you do...
No, I only drink when I'm thirsty.
Yeah. You basically...
And I drink as much as water is necessary to kill my thirst, and that's usually rainwater below 125 ppm. It's very simple.
Got it. And
you have thirst.
Are you sourcing that in a specific way, or are you mixing that with deuterium depleted water?
It's deuterium depleted, but it's in the rain, kind of a good fall rain, 125 ppm or below. That's practically, tap water is 143 ppm to 155 ppm. Most of the major cities, if it's coming from higher elevations, for example, the Colorado River, it may go down to 138 ppm, but it's still very high when you drink 2 or 3 liters of it a day.
Yeah. So that's
the
what I
problem
wanted
is,
to get into, the elevation and then the latitude, right?
I tell you, with my buddies from my church, we went up to the Carpathian to take a walk. We walked up to like 3,000 meters. And so as we arrived at the parking lot, my friends, they came with a backpack with a kind of a water sipping.
I hate those.
They actually, and they were asking me if I had any water. I said, no, I don't. And they said, well, it's going to be an 8 hours walk up and down. I said, yeah, that's fine. And I ate my 2 or 3 pieces of bacon before that. And I just put my boots on and we just walked.
Yeah.
And so they were asking me if I'm like want to drink of their water. I said, well, listen, if I'm thirsty, there's this beautiful mountain river, the stream coming down. That's where all the living creatures drink here. I consider myself one of them. So if I'm thirsty, I just kind of, if I'm that thirsty, I kind of like
pushing
a
the
little,
limit. You like to push the limit, the challenge.
I consider everything that you put in your body as a deuterium source, potentially. So you have to kind of, if you don't know the deuterium content, you have to limit the intake. So for that matter, and the other problem is with excessive water drinking without salt is that osmolarity will drop in your blood. Osmolarity will drop in your brain cells. So you will actually have brain swelling that dismantles the hypophysis, or the pituitary gland it's actually locked into your sella turcica, which is a bony little compartment. That's where this pituitary gland sits in. And if you drink water excessively, this gland kind of expands in this bony capture, or it's capsized in this bony compartment called sella turcica, or the Turkish saddle for that matter, because it looks like one, and actually start compressing on ADH releasing cells, the antidiuretic hormone related, and also sex hormone and growth hormone and thyroid hormone regulating cells, because they are all produced by the pituitary gland. So if those are swollen, then your antidiuretic hormone is not helping you to preserve water, so you develop diabetes insipidus, and you have to drink more because you are more thirsty, simply because you are unable to salvage water through the actions of antidiuretic hormone, which prevents diuresis. It actually retains water from your primary filtrate in your kidneys. So you cannot regulate your own water homeostasis. So you're actually exposed to a environmental, unpredictable deuterium source in the form of water and causing brain. Actually, you can actually die of water poisoning. Mm-hmm. You can die of brain swelling. In New Jersey, I believe early summer, there was a mother who took the kids on a mountain walk, and she drank about 1.5 liters in 15 minutes. By the time she drove the car back, she fell into a coma and she died in the garage.
Wow.
So it's excessive water drinking without thirst is causing more harm than you can actually imagine. And it's simply because of your body has your thirst or any other natural signal. For example, if you're hungry, you eat. If you're tired, and then you sleep. If you're thirsty, then you drink. If you are kind of, and sexuality is controlled pretty much the same way. So we have these basic instincts that's practically just, you don't go to sleep simply because you see a sofa bed somewhere. You don't eat just because you walk by a restaurant. You don't eat just because it's available. You don't, and you shouldn't be drinking just because there is bottled water in the department store. And you heard on the radio that you should drink 2 or 3 liters of water because it's a good business for the water industry, the bottling water, the bottled water industry. Actually, if you drink excessively, let's say a liter of water in half an hour, you're gonna pee out 1.3 liters of water in the next 4 hours for sure, but it's gone in the next 2 hours practically. So it's useless. Your body is trying to get rid of it simply because it didn't need it. So you are constantly challenging your body to kind of overcome your behavioral patterns simply that are physiologically, biochemically, totally against your own regulatory systems. Those are driven by media, by advertisements, and the list just goes on. And that's our kind of scientific position on this whole issue.
Yeah, I love it. I mean, I'm the same way. I like when hunting in Wyoming, and I actually forgot to bring water at all. And I was just like, I just I had my water bottle. I'll just fill up at a stream if I get thirsty. And it's not that hot either. So I have buddies I've done these big hikes with. They all bring the Camelbacks and they just get comfortable. They get used to just sipping on the Camelback, the water or backpack, like every 5 minutes. They're like, it's just something you psychologically get used to. And, the same people, they have their big jug and they just sipping. It's just like they get used to that. So that's that feedback loop they're in. And it's hard to break, but I challenge everyone. I think, first off, drink water that's actually alive, spring water, and then something that you don't really need as much of. If you challenge yourself, obviously, if you're in a sauna or you're really 35 C out and you're sweating profusely, it's a different story. But every day, we're not really sweating that much, needing to drinking that much
listen,
water.
if you're thirsty, drink as much as it's necessary. When we were kids, we went out on these school trips. And I remember teachers telling us when we were like, I'm thirsty. I said, just don't drink. Don't drink. If you're still thirsty in 5 minutes, wait 5 minutes. Then you get one little sip. That's it. And actually, it was sufficient just to, we were lean. We were not like as obese as nowadays these kids. Unfortunately, if you look at the average body composition, I still have 14%, 15% fat content of my body. And
pretty good.
I just keep these, if I go to I'm 62 now, and if I go to a reunion, God, I mean, some of my friends just like, okay, actually we're going to go and say, the final goodbye to my guitar player in high school. We started, and same age, and we're gonna bury him on Friday. Oh. And I mean, just one problem after another.
It's really sad, and the U.S. I think is even worse than in Hungary or in Europe. But
unfortunately, we are getting there.
Yeah, it's all on the same trajectory, really. But something that, you know, is easy to do. But again, Laszlo, people are not going to, they want the pill, they want the easy fix. I mean, most listeners to our show know that the lifestyle interventions, the decentralized health model is the most effective. Obviously, it's the longer lasting solutions. There is no Band-Aid or supplement that's going to provide you this.
And it's very important, Tristan, what you guys do, especially for your age group. I'm an old guy now, so I don't really have as much as like think ahead of you guys do as far as health. I mean, knock on wood, I'm okay. I'm 62 and I'm happy it's that way. And I wish everybody would be in this situation. I know how to kind of preserve this option or possibility. It's all through food and lifestyle. There's I don't take any supplements.
Yeah.
I don't take any special remedy for, you know, preserving this type of, you know.
Yeah, no. And that's how I am now too. And I was just going to ask quickly, because the deuterium depleted water is, you know, it is accessible for people. And I know they have as low as, I don't know if it's 10 or 20 ppm, and you can use that to mix to like 100, 120, maybe 130. Is that something you advocate for, you know, the everyday person trying to be healthier if they can't get, let's say, a lower deuterium content water? Or do you really only see that as kind of a medical intervention for those, say, with cancer or with a serious health issue?
Yeah, there are therapeutic applications below 100 ppm.
Okay.
Yeah, that for those you need to go to a integrative setting, and you need to talk to doctors who are familiar with this deuterium story. And for that matter, it's practically just a lifestyle. The safest water is what you eat, is not what you drink. And your body makes the safest water for you. How to deplete water the easiest? Just eat, you know, grass-fed animal fat. Because I get a lot of questions is like, is rainwater, can I set up my own deuterium depleting machine? Can I do this? Can I do that? I'm like, listen, no, just go to the grass-fed food store, get some fat, and that's your fastest water. So you don't have to buy anything.
It's cheaper.
Just
too.
eat.
Yeah.
It's much cheaper. It actually makes you like live free of this establishment. You don't have to go to doctors. You don't have to go to clothing stores. You don't have to buy medicines. You don't have to spend time in doctors' offices. You don't have to drive there. You don't have to pay. It's much cheaper after all, if you just look at this whole scenario.
That initial investment, you know, for that low time preference. But yeah, that's fantastic. I guess, you know, it always comes down to the same things. And that lifestyle habit really drives your health outcomes. And at the end of the day, I think adding this deuterium message is on top of everything that we talk about as well. It aligns very well. But I'm excited to see more and more this research come out, and, you know, read your paper on the mountaineering as well. It's all related. It all comes back to mitochondrial health, I think, at the end of the day. So yeah, I guess, you know, wrapping up, what are you most excited about to research or coming to fruition in this space, or other aspects maybe of quantum biology, or kind of this frontier of research and in the fringe, I guess?
I'm kind of in a interesting situation, because I do a lot of editing work for scientific journals, and I invite reviewers and deuterptation is practically the deuterium angle interpretation of medical data. This is what we do now in many of those papers. I get lots of data. I have Stephanie Senev, Antonis Kyriakopoulos, if I say it right. He's a Greek physician, colleague and friend of mine. And I work with a number of other scientists, especially when there's a need for it. And I actually get a lot of questions, data that I can position, and I can kind of explain from the deuteronomics angle, and that's fun. I mean, it's serious scientific work, and it requires a lot of background biochemistry knowledge, and there's a lot of discovery. I love corresponding with Dr. Roman Zubarev, who is at the Karolinska Institute. He published a paper about the gray seals and the peregrine falcons having 315 ppm proline-based deuterium accumulation in their collagen. It's practically the physical forces they have to endure. It's a phenotypic data. It's in the Journal of the American Chemical Society.
I just wanted to ask about proline real quick because I forgot. I've listened, I think it was Stephanie Seneff who mentioned that proline can it traps that, and that aligns with what you're saying is once proline has deuterium in its structure, it's not going anywhere. Is that a good or like a bad thing?
Yeah, I started kind of delivering this paper and information to Dr. Seneff and so on, so we can actually get her expertise and so on. Many of these dots are kind of the first little drawing is started by me simply because I'm they look at me like the go-to guy when it comes to deuteronomics. So, and I'm very happy to work with everybody because every kind of different angle, look, eyes just help to uncover all these stories. And Dr. Seneff, she made a lot of contributions on her side. And actually, she knows more about proline-related deuteration than I do now, even though I started this type of discussion as well. But she dug into this. We together did, but I have not reviewed yet many of the materials that we did collected, and most of those come from her searches. So her contributions are very critical and fundamental and very original to this matter. We don't know all the details yet to give you kind of very insightful, easy to understand comments or opinion, but maybe in a few months we'll be able to cover that as well. But ask Dr. Seneff because she will have very clear answers to some of these
and it really just goes along with, I think maybe the question to her was like, how can you clear out like high levels of deuterium? But the way, the mitochondrial turnover is so high, it's like if you change your diet, you're probably going to be getting deuterium depleted water out of the mitochondria pretty quickly. So it's not something, and maybe she was tying that into glyphosate anyway as well. So I'll definitely ask her about that. But I guess
they are all connected because glyphosate destroys oxygen carrying capacity of red blood cells. Then mitochondrial oxygen supplies are not sufficient to produce enough metabolic water. There's deuterium depleted water. I mean, I can go on, and we can do this.
We can do it forever, and it's just so interesting. But
it's very interesting.
The last question I have is how is the research, I guess the funding for all this going? Because I know that's a struggle in more of these alternative research spaces that aren't very profitable. And it's something I'm interested in maybe helping out on or trying to find a way, because there's things like...
Listen, I stopped applying for grants, scientific grants, in 2003. Some 32 grant proposals were triaged by the NIH, the National Institutes of Health, by those quote-unquote reviewers. It's a club of people. If you kiss enough ass around, then I guess you can get your funding for some bullshit studies. So, I mean, that's what oncology in general. I'm just giving you a broad picture of how it goes. And I was working as a subcontractee of those big funded projects because we could provide tracer-based, 13C isotope-based, deuterium-based, very precise metabolic profiling scenarios. And so we got our funding indirectly. They never funded directly our research, but indirectly we could. We published over 100 papers in the peer-reviewed medical literature. We are very well established using these scenarios. And if you are not greedy, meaning that a Nissan Sentra is okay, you don't have to drive a Porsche, and you don't have to have a, who knows, big size of a house, or you just want to rent. Like I'm really not, financially, I'm not interested in science. I'm interested in science because of science itself. Mm-hmm. And that saves you a lot of hassle. So I stopped wasting my time on applying for NIH funding because I knew those guys will never fund my, studies, my work, because, I'm not an easygoing guy. I do state my case, and I like to be criticized, and I like to answer questions, and I'm very happy to work with people, very happy to take challenges. And I do my consultations, $80 an hour. Some other doctors do $500 an hour. Because I just want to help people, but I still have to kind of run my operations. And in the meantime, I'm very happy to collaborate in any given, build these laboratories and help people to achieve their health by science and knowledge and education, and deuteronomics to be in their agenda sooner or later. But it's practically an educational process that pays the bill. If you don't do this for money, but you do this for science, and you do this just simply contribute the best way you can, then you're okay.
Well, I want to thank you so much for your work. And yeah, if there's anything people can do to support your work or the work of deuteronomics research, it'd be great to let them know. Definitely, you can keep me updated on the papers and everything, and let's stay in touch. But yeah, how can people support you or find out more about your work? They could probably just Google your name and your research. But is there a specific place you?
Yeah, if you type in deuteronomics or deuterium and my last name Boros, or you just go to LaszloGboros.com. It's my first name, my middle initial, and my last name. No dots, no separators, just LaszloGboros.com. You're going to see a lot of blogs, podcasts, scientific papers, previous and current ones. And you can book a consult if you have specific questions. I have an hour set aside for $80, and you can just kind of ask anything you want. And if I have an answer, I'm very happy to share it with you.
Well, that's fantastic. Thank you so much for coming on and sharing your time. And this was a lot of fun. Let's definitely stay in touch. And I'm definitely going to be talking
I'll send
more.
You that paper, the draft, so you can read it before it.
Amazing.
The review
I love when researchers
just don't share it with anyone, because
yeah.
This is right now. It's just practically because we talked about it. I think you should look at it.
Yeah, I would love it. Mountaineering and hiking is my jam, and yeah, it's exciting stuff. Excited to talk to Stephanie as well, and yeah, thank you so much, Laszlo. This was a pleasure, and really appreciate you coming on.
Thank you so much. Have a good day.
Thanks, everyone. We'll see you next time.